This video and the ones after it are intended to help you review for the Unit 1 exam at Lane Community College. This review is patterned on a live review session I had with the Tuesday-Thursday afternoon section that went pretty well, so I'm just going to do that. And the way it will work is I will tell you some things, and then other things I will ask you to answer some questions, and I strongly suggest you pause the video and answer the questions when prompted. That will help you assess how ready you are. As always, I want you to go back to the slides to get information, watch the videos to get information, and use the study guides where possible. And that will help you prepare. In terms of what words to know and things like that, I think in general a good rule of thumb is if a word has come up in lecture, so if it's in your notes or if I talked about it a lot in a video, then you should probably be willing to or be ready to write it in a fill-in-the-blank question. But words that I gloss over, if I don't specifically tell you to learn them, then you might learn those last. So, or just be ready to match a definition to them or something like that. That's always a good rule of thumb. Okay, so let's get into the review. So we started with lecture 1, and the main topic here was the central dogma of molecular biology. And so this was proteins and nucleic acids and how proteins are made. And what is a protein? You should be able to tell me that. So pause the video and tell me what a protein is. A protein is a chain of amino acids, and the order of the amino acids is important. That order is determined by what? Pause the video and tell me. So that the order of amino acids is determined by the order of nucleotides in the messenger RNA molecule. And that was determined by the order of nucleotides in the DNA of the gene that encodes the protein. So that's the basic idea behind the central dogma, is proteins are important, and the information for making a protein is stored in DNA, and as an intermediary we need a messenger RNA molecule. DNA and RNA are made of nucleotides, and they are basically identical. There are some chemical differences, but the information they contain can be identical. Proteins are very different, but they're based on the nucleic acids. What you need to know about that is all the things I've said, and again, go back to what are the differences between DNA and RNA, what are the differences between DNA, RNA, and proteins, but also what are the parts of a cell that make RNA, what makes proteins, what is it called when we make proteins, what is it called when we make RNA. Make sure you are looking at those things. And then we moved on to microscopy. And in microscopy, there were really just 3 big words we cared about. And I want you to be able to tell me these words and tell me what they mean. Oops, so, hello, come on. So, what those words are going to mean, I'll go into it briefly, but the words are magnification, resolution, and contrast. And my terrible handwriting can be improved by your stopping and spelling things correctly. So magnification and resolution are things we might understand pretty well. Magnification is easy to do with lenses. You can stack lenses on top of each other to increase magnification. You can shape a mirror more grotesquely to make it magnify something more. You can use electronics to make pixels much bigger and magnify something. But resolution has physical limits. There is a limit to the information that can be carried by light at certain wavelengths. And we see that with telescopes, where a bigger mirror gives us better resolution. That's why we have to have a huge James Webb Space Telescope. It has a huge mirror, and that is to give it better resolution than smaller telescopes can get. And that is just a physical reality. A smaller telescope cannot pretend to be a bigger telescope. They can have the same magnification, but the bigger one has better resolution. And then it's the same with microscopes. The microscopes in the classroom are already at the limit of resolution. We cannot get better resolution than what they already do. So that's just a thing to understand. And then contrast matters because cells and tissues don't have very much of it usually. And so when you do microscopy, you have to think about, how will I see anything through this microscope? And generally we have to use stains or pigments that give cells color, or we have to use a microscope that generates contrast for us. So be aware of those words. And then that brings us to cell structures of prokaryotes. And the place we start is compare them to eukaryotes. So pause the video and list some things we find in eukaryotes that we do not find in prokaryotes. So things we find in eukaryotes include nucleus, endoplasmic reticulum, Golgi, mitochondria, chloroplasts, lysosomes. That's a pretty good list. Those are all membrane-bound organelles that we do not find in prokaryotes. We don't find them in bacteria. We don't find them in archaea. What we do find in all three of those types of cells on the inside, what do we find in every cell? What's in every cell? DNA is in every cell. RNA is in every cell. Proteins are in every cell on Earth. That matters for a lot of reasons. RNA and proteins we can attack with antibiotics and disinfectants. Lipids. Every cell on Earth has lipids in a cell membrane. We can attack that with disinfectants and heat. We can attack proteins with heat. Antibiotics some of them go after proteins. Equally important, viruses try to hijack the system cells use to make proteins. So it's important to know these things. Another thing I mentioned in class is that you'll have to draw a gram-positive or gram-negative cell. Just to reiterate, the level of detail I want is something like this, where you draw the cell membrane, and then if the cell is gram-positive, you draw a thick peptidoglycan layer around it, and you label it peptidoglycan. So cell membrane, and then peptidoglycan. I'm rusty. I haven't taught remotely in a long time, so I'm not good at drawing on this thing. I apologize. So, and then for gram-positive, you'd have to write, you'd have to draw in and label teichoic acids. Why are we doing this? There are a couple different reasons. One is to remember peptidoglycan is on the outside of a gram-positive cell, where enzymes like lysozyme can get to it. So lysozyme can kill gram-positive cells. Teichoic acids and peptidoglycan are both detectable by our innate immune systems. And a macrophage is likely to bump into a teichoic acid or to peptidoglycan. And once it does that, it knows, hey, this is a gram-positive bacterium. I'm going to eat it. If I ask you to also write something that's inside the cell, you could write DNA, RNA, proteins. But another reason we do this is to know what not to draw. For gram-positive, you do not draw a lipopolysaccharide. You do not draw an outer membrane. You do not draw, well, that's a pretty good list, but you don't draw a nucleus or endoplasmic reticulum or any of those other things. So make sure you know, what would you find in a gram-positive cell and how would you draw it? What would you find in a gram-negative cell? How would you draw it? What would you find in a eukaryote? How would you draw it? That's the important stuff from those structures. But why does it jump back to the beginning? But there are other structures to look at. There's chromosomes and plasmids. Those are both DNA molecules. And the idea there is, let's use galaxy. So the idea is a chromosome is a big DNA molecule all curled up inside of a bacterial cell. So think of it the same way you think of a chromosome in a human cell, except instead of 46, there is 1. That's a chromosome. And all the genes on here are for things like how to make proteins, how to make regulatory proteins, how to make structural proteins, how to make enzymes, how to make everything the cell needs to divide. A plasmid is a much smaller DNA molecule with a lot fewer genes. So a plasmid has genes for non-essential things, or things that are only essential in some rare situations. And we care about this a lot because cells can share these. The cell can give a plasmid to another cell. That means if this plasmid has instructions for how to cause a disease or how to resist antibiotics, the cell can share that with another cell by giving it the plasmid. So we care very much about what are plasmids. Okay, that's those things. Let's see, what else? Yeah, other structures, flagella, what do those do? Fimbriae, pili, glycocalyx, those are all good structures to know. And that's really a recap of Unit 1, not Unit 1. That's really a recap of Lecture 1. In the next video, we'll move on to Lecture 2.